Hi everyone, welcome you all to the next session on Exam Focus 2023. So we'll be dealing with Enamel and we'll have a short glimpse of the entire chapter. I'll be dealing with a little bit on introduction of enamel, properties of enamel, the basic structure and all the other structures found within the enamel, clinical considerations and changes of enamel. So when you look at enamel, what exactly is it? It is a hard mineralized tissue and which forms a protective cover on the surface of a crown. So it is present only within the crown portion and it is a mineralized epithelial tissue or it is a product of the epithelium. While the remaining others like dentin, pulp, cementum, your alveolar bone, periodontal ligament, all those are actually connective tissue structures. So only enamel among the entire tooth is a part of the epithelium. And the mature enamel is actually acellular. That means it does not have cells residing within the enamel. So it is considered as an acellular structure as well as an avascular structure. So we'll go on to the properties of enamel. So we'll deal with both physical and the chemical composition of enamel. When you look at the physical properties of enamel, you have certain properties like hardness, brittleness, the density, the thickness of the enamel. So all these are considered as the physical properties. Hardness, enamel is considered to be very, very hard because of its high mineral content. And this is required so that it can withstand a lot number of forces which fall on the enamel. And the hardness decreases from the surface as you go towards a dentino-enamel junction or from the cuspal tip to the cervical margin. So the hardness will keep decreasing as you go inwards. And when you consider the brittle nature of enamel, it is highly brittle. And it is the underlying dentin which is more viscoelastic, which is able to support the enamel. Otherwise, enamel will fracture when excess force is applied. Density, again the density decreases from the surface of the enamel to the dentino-enamel junction, and permanent teeth is actually considered to be more dense than the deciduous dentitions. When you look at thickness, again the thickness varies from teeth to teeth. It is the molars which has got the maximum thickness, especially the molars is around 2.6 millimeter, and as you go towards like incisors and all, the thickness decreases. And when you check for the thickness of the enamel, the thickness is maximum in the cuspal region and it is very thin at the cervical region. The other physical properties include the color of enamel. So usually the enamel is transparent or translucent, but the opaqueness varies. So if it is more opaque, if the enamel appears more opaque, then the color will be either grayish or bluish white. And if the enamel is very, very thin, so the opaqueness will depend upon the thickness of the enamel. So if enamel is very, very thick, then it will look more white in color. And if it is very, very thin, it is the underlying color of the dentin that is going to be reflected. So it will appear a little bit yellow in color. Again, when you look at the permeability, enamel is considered as a semi-permeable membrane. And as age advances, the permeability will keep decreasing. That is because of the changing enamel matrix. When you look at the refractive property of the enamel, it is fully made up of crystals. So enamel is completely made up of minerals or crystals. Because of that, it will have a reflective, light reflecting property. So it is birefringent crystalline nature, or it has a birefringent crystalline structure. If you keep it under polarizing microscopy, it will show varying colors. It will show red color, green color, blue color, and the refractive index is around 1.6. And another thing which you have to remember is enamel cannot undergo repair or it cannot be replaced. So if once enamel is lost, it's going to be lost forever. When you look at the chemical composition, enamel consists of three portions. So it has inorganic component, it has water in it, and it has organic component. And the inorganic component is around 96%. That is the minerals. Water is around 3% and organic content is just 1%. When you look at the inorganic portion, it is basically made up of hydroxyapatite crystals, which is made up of calcium, phosphate and hydroxyl ions. So these three ions constitute the main crystal. So this is the formula for a unit cell. That is, a single hydroxyapatite crystal is going to be made up of this formula, that is calcium 10, PO4 6 times and OH4 twice. And when you look at the structure of a hydroxyapatite crystal, you have in the center an OH ion and surrounded by calcium and phosphate groups arranged in triangles around the OH ion. That is how it is a hexagon, and in the hexagon you have alternating arrangement of calcium and phosphate with the central core of OH ion. So this is called as a unit cell, which is a basic unit of enamel. Water, the necessity of water is to provide hydration to these crystals, and also it will facilitate the transportation of molecules across the enamel. Organic content is just 1%. Out of that, 0.4% is your enamel proteins, and the remaining 0.46 is consisting of carbohydrates, lipids and other organic substances. The organic portion of enamel is basically made up of enamel proteins, and these enamel proteins are of two types, that is amelogenins and non-amelogenins. So when you look at the composition of a matured enamel, it is just 0.4% which is going to be your enamel proteins. So in fact, the ameloblast secretes 100% of enamel proteins. So your organic matrix initially which is secreted is completely of this enamel protein. But in a matured enamel, you will have just 0.4% of the entire enamel proteins which is being secreted. So it is made up of two types, that is amelogenins and non-amelogenins. Now amelogenins are actually low molecular weight protein which forms around the majority among the enamel proteins. It forms around 90% of the enamel proteins, and it consists of amino acids like proline, histidine, leucine, glutamine, and this amelogenin is actually located on X chromosome, especially your X chromosomes. When you look at the other enamel protein, it is actually non-amelogenins, which is a high molecular weight protein which forms a lesser amount among both the enamel proteins, and they are rich in amino acids like glycine, aspartic acid. The different types of non-amelogenins, it consists of tuftelin, enamelin and ameloblastin. Now all these enamel proteins, they have a role to play, and once the role is done, they usually leave from the matrix. So their main role is to help in mineralization. So they play a role in mineralization. Ameloblastin will act as a calcium binding protein. Enamelin will help to strongly bind all the minerals together, and tuftelin will help in the initial mineralization. Next, we move on to the structure of enamel. So, what exactly makes up the enamel? That is a basic structure of enamel. The basic structure of enamel is formed by three things. It consists of enamel rod or prism, which is this. Then you have something called as an interprismatic substance. That is, between two prisms, you will have a mineralized area which is called as interprismatic substance. And you have a third thing which is present surrounding the enamel rod, that is going to be the rod sheath. So enamel rod or prism is a basic fundamental unit structure of enamel. So the entire enamel is made up of crystals or prisms or rod. The crystal is basically hydroxyapatite crystal. And the interprismatic substance is also made up of crystals. So it is between two rods you get this interprismatic substance. Now the difference is that there is change in the direction of these crystals in this area. So when compared to the rod, the interprismatic area, the rods are all in different direction. Rod sheath, sheath means outer covering of the rod. So it is an outer covering which is present on the outer surface of the enamel rod. This is an organic matrix. So it is made up of organic components. This space what you see here is your rod sheath. Then when looking at the other things of enamel rod, like basically the shape, the arrangement of crystals within the enamel rod, how many rods are there. So the shape of the enamel rod is actually on a longitudinal section. It appears like a cylinder. This is how you see, and it is not a typical cylinder. It's more of a flat thing. That is, it has a slate like appearance. It is more flattened. It's long, but it has a slate like appearance. And on a cross section, when you take the cross section, that is the cross section of the cylinder, it look like a hexagon. So in a cross section it is hexagon. In a longitudinal section it appears like a cylinder, or it has a slate like appearance. And when you look at the arrangement of crystals within these prisms or the rod, it is tightly organized. So if you see, they are very tightly packed, and the crystals are tightly packed within the enamel rod. And if you look at the direction of these crystals, in the center portion of the rod, it is parallel to each other. But when they go out towards the periphery, it becomes flaring laterally. That means it will start to be arranged in an oblique manner. The enamel rod is wavy in nature. You can see in this picture, it has a small wavy nature, and the number of enamel rods within the tooth varies between 5 million to around 12 million. And the length and the diameter of the rod, in the cuspal region, the rods are usually longer, and the rods are much longer because of their wavy nature. So if you look at the height of the crown, or the length of the crown, the width of the crown, the length of the rod is going to be greater than the height of the crown, that is from DEJ to the surface. That is because of the wavy nature, it will be actually more longer. And so the enamel rods are longer at the cuspal area, and it's going to be shorter at the cervical region. And when you compare between the length and the diameter, usually it is 1:2 ratio. So between DEJ to surface, if you look at the diameter of it, at the DEJ, the diameter of the rod is 1, and at the surface, it's going to be 2. So as a rod appears to be on the outer surface, the diameter is going to be much more greater than what is seen at the dentino-enamel junction. The other things which you have to look into enamel rod is the pattern of arrangement on the rod. So enamel rod, usually if you take a cross section, it appears like a hexagon, or it appears like your fish scale pattern, how you see the scales on the fish. And also the pattern of arrangement. The pattern of arrangement is going to be different for these enamel rods. This is a pattern 1, where the interprismatic substance is larger or greater, or there is distance between two main rod area, or the interprismatic substance is going to be greater. This is how it appears in pattern 1. And you have patterns like staggered, stacked, and irregular. Staggered is irregularly arranged but in rows. Stacked is when it is neatly arranged in rows. And the main pattern which you can see, like I said, is a keyhole pattern, or it is considered as a fish scale pattern. It appears like a keyhole. So it is called as a keyhole pattern. And when looking at the direction of enamel rods, it usually runs from the dentino-enamel junction to the surface. And at the cuspal area, it is almost vertical. So almost 90 degrees at the cuspal tip. Towards the mid portion of the crown, it becomes oblique. And as you go towards the cervical region, there is a slight difference in the orientation of the rod in deciduous dentition and permanent dentition in the cervical region. So in the cervical region, it is different. You look here, in deciduous dentition, it is horizontal, while in permanent dentition, it is directed towards the gingival region, or it is directed towards the apical region. The direction of the rod is towards the apical region. So there is a difference in permanent and deciduous, especially at the cervical portion of the enamel. And rod arrangement, it is usually in rows. So if you see the crown, it is going to be something like this. And each area, each part of the crown, it is arranged as rows. And it runs around the entire circumference of the tooth. So that covers the basic structure of enamel, that is rod, interrod and rod sheath. So about the rod, you have to talk in detail about what is the shape of the rod, how is the nature of the rod, the direction of the rod, the number, the length, the diameter, all that you have to discuss under enamel rod. Now the other one which you can see is something called as a prismless enamel, or it is otherwise called as aprismatic enamel, or it is called as rodless enamel. So aprismatic enamel means absence of prism. You don't have rod and interrod, like the structure you saw before. Rodless enamel is absence of enamel. Now this is a structureless layer. It is a structureless layer which is 30 micrometers thick, and this is seen at the dentino-enamel junction and on the surface of the enamel. So prismless enamel or aprismatic enamel is seen on the surface of the enamel and at the dentino-enamel junction. This is a highly mineralized layer. So that means only thing is that you don't have a particular arrangement of rod and interrod, but the area that you see is going to be highly mineralized. And here you have crystals, but the crystals are arranged parallel to each other and they are perpendicular to the surface of the enamel. Now, why do you get a structure like this? Why is that you get an area without rod and interrod? That is because of the change in the morphology of Tomb's process. So, if you remember, it is a Tomb's process. After the formation of Tomb's process only, you will get rod and interrod. So, at the dentino-enamel junction, when the initial layer of enamel is secreted, you don't have this extension. That is why you get an area where there is no rod and interrod. So it results as due to the elimination of Tomb's process. So this is how you get, this is a highly mineralized layer, and this can be asked as a 3-mark question. Prismatic enamel, aprismatic enamel or rodless enamel. The other structures which you see in enamel, that includes incremental lines. So these are light microscopic structures which you see within the enamel. Under the light microscope you can see them. They are other structures other than your enamel rod. So it includes incremental lines, Hundertschrieger bands, enamel lamellae, enamel tuft, enamel spindle, gnarled enamel and dentino-enamel junction. So these are the structures which is seen apart from your basic structure, that is the enamel rod. Now we'll go into detail of each. Now what are incremental lines? So these lines represents period of activity alternating with period of rest. So basically an incremental line is formed when the ameloblast will take rest. So after period of activity, after depositing the enamel, it will take rest, and that period of rest is considered as your incremental line. Now remember, enamel is secreted in increments. It is not together at one stretch that enamel is going to be secreted. It is deposited as increments, and an increment per day. Per day enamel is going to be secreted as 4 microns per day. So you have 3 types of incremental lines that include, one is cross striation, the other one is represented by incremental lines of Retzius or striae of Retzius. Now cross striations represents daily activity. In a day, how much the ameloblast works and how much the ameloblast rests. So that is daily activity is represented by cross striations. That is after 4 microns per day, after it secretes, it will take rest in a day. So that will form one single cross striation. And when you look at the direction of these cross striations, they are lines traversing at right angles to the long axis of the rod. So if you see this picture, you can see perpendicular black small lines which is perpendicular to your enamel rod. That represents cross striations, which is a resting period of the enamel in one day. Striae of Retzius is a weekly resting line. That is long periodic line, that is following a week the ameloblast will take a long break. That is considered as incremental lines of Retzius or striae of Retzius. So these striae will appear as brown color bands in a ground section. This is how it appears. These brown color lines, what you see is your incremental lines of Retzius or striae of Retzius. So remember, it is a weekly resting period of the ameloblast, which is of a longer duration rest. It will take rest for a longer period. That is why it is called as a long periodic line. And so the ameloblast will usually work for around 4 to 5 days. That is the working period of an ameloblast. So the distance between two striae of Retzius will vary between 16 to 20 micrometers. That is because into 4 days or into 5 days. 4 into 4 or 4 into 5 days. And when you look at the arrangement of these striae of Retzius, if you see, notice here, around the tip of the dentin, it is arranged as semicircle. This is how it is arranged around the tip of the dentin. And as you go downwards, it becomes obliquely arranged. So from the DEJ to the surface, it will be obliquely arranged and it is deviating occlusally. So the direction of the enamel rod is towards the occlusal surface. And when you take a cross section or a transverse section, this will appear like the growth rings which you see on a tree. Then there is another incremental line which is basically the striae of Retzius itself. It's called as a neonatal line. So it is the striae of Retzius itself which we rename as neonatal line. That is because when there is a change in the environment during enamel deposition, that is prenatal and postnatal. So intrauterine and outside the uterus, when there is a change in the environment, ameloblast gets affected. And during that period, to adapt to the change, the ameloblast will take a longer rest. So that resting period, a longer or a bigger striae of Retzius, accentuated striae of Retzius, is called as a neonatal line, which demarcates between your prenatal and postnatal enamel, which is seen in all deciduous teeth and also in first permanent molars. The other structure what you can see is Hunter-Schreger band. Now, Hunter-Schreger band is basically enamel rod itself, and you know that enamel rods have a tendency for reflective property. So this brings around an optical phenomena. So it is this Hunter-Schreger band is produced as an optical phenomena. And what you see is series of alternating black and white color bands. If you look at this picture, it's very clear. You can see white bands and black color bands, and it is alternately arranged. And this is seen under reflected light. Now, why is that you get such a band? That is because there is a lot of change in the direction of these enamel rods. There is an abrupt change in the direction of these enamel rod, which will produce such an optical phenomena. You look at the location, they arise from the dentino-enamel junction and they move towards the surface of the enamel. So they extend up to 2/3 of the surface of the enamel. And the names of these black and white color bands. So the black color bands is diazone and the white color bands is parazone. And the angle between the two is considered as 40 degrees. So remember, Hunter-Schreger bands are produced as a result of optical phenomena. They are made up of alternating white color bands and black color bands, where the black color is called as diazones and the white color bands is parazone. White occurs because of change in the direction of enamel rod. Where it is seen from dentino-enamel junction outwards towards the surface, and it runs till 2/3 of your enamel. The other structure which you can see within the enamel is enamel lamellae. Now, enamel lamellae is a thin leaf-like structure which arises from the surface and extends towards the dentino-enamel junction. How is that you get this? This develops this kind of lamellae or a hypomineralized area develops when there is too much of tension or extra stress. And this is made up of only organic matrix. So this area what you see is made up of organic matrix. Now what is the importance of this? It has a clinical importance because it is extending from the surface inwards into the dentin, and it is organic matrix. It is easier for the microorganisms to enter into the dentin. So it provides a pathway for the entry of microorganisms leading to progression of dental caries. Then you have three different types of lamellae. So the lamellae is divided into three types: A, B and C. A is basically typically made up of poorly calcified enamel rods. Your B is made up of poorly calcified enamel rods and degenerated cells. C is completely made up of organic matrix, and this is seen mostly in erupting tooth. So A is only found within the enamel, while B and C crosses the dentino-enamel junction into the dentin. So B and C reach up to the dentin, while A is only present within the enamel. So that is enamel lamellae. Enamel tuft is basically narrow ribbon-like structures, or they resemble tuft of grass, how your grass appears in bunches. So this is how it's seen. It is seen at the dentino-enamel junction. So they resemble tuft of grass, or they are narrow ribbon-like structures which actually can branch. If you see, you can see branching of these small ribbon-like structures. Now, how is that they are formed here? How is that you see this? Again, they are actually due to abrupt, sudden changes in the direction of rod. Because of that, you get this. So, there is an abrupt or a sudden change in the direction of rod. And they are basically made up of poorly calcified enamel rods. So, they are enamel rods itself, but they are poorly calcified enamel rod. Because of the change in the direction, there is going to be the calcification of the rods is going to be affected. So it arises from the dentino-enamel junction. It is seen up to 1/5 or 1/3 of the thickness of the enamel, and it contains organic matrix, especially an enamel protein called as tuftelin. The other structure which you see is enamel spindle. Now, enamel spindle, you should remember that this is the only structure which is actually not a part of enamel but seen in enamel. That means it is a part of dentin. It is a structure which is a part of the dentin, but it is going to be present within the enamel. So, only structure in enamel which is truly not a part of enamel but it is seen in enamel. So, this is actually a slender projection, a small projection which bypasses your dentino-enamel junction and extends to the enamel. So this is made up of odontoblastic process. So it is the extension of odontoblastic process into your enamel across the dentino-enamel junction. And this, what happened is, how do you get this odontoblastic process within the enamel? It would have got trapped during amelogenesis. So the odontoblastic process would have got trapped in between the enamel matrix during amelogenesis before mineralization. So if you see, this is what it is. It is a small comma shaped structure which is thickened at the end portion. You can see the thickening at the end of it. Okay, and this is perpendicular to the dentino-enamel junction. And when you take a ground section, it appears black in color because when you're making a ground substance, since it's an organic matrix or it is odontoblastic process, it will disintegrate and it's going to be replaced by air. The other structure is gnarled enamel. Gnarled enamel is basically seen in cuspal region. It is seen in the cuspal region. How is you get gnarling of enamel? So it is basically intertwining of enamel rods. It is the intertwining of enamel rods. That is because in the cuspal area, it is narrow. There is not much of space. So due to lack of space, the ameloblast, when they are moving towards the surface, they crisscross each other. So the amyloblasts will cross over each other, and that is how you get intertwining of enamel rods. Now, though it is because of the lack of space, it has a function to be served, because when you have a rope which is intertwined together, that rope is going to be much more stronger. So similarly, when you have enamel rods which are twined with each other, which are twisted around each other, that will add more strength to the cuspal region. So one of the functions of knurled enamel is it will provide strength and it's able to resist fracture. Again, this is an optical phenomena. So if they ask you a question, so any of these structures which you studied, that is incremental lines, which is made up of two lines and one more line, that is striae Fritzius. You have lamellae, tuft, spindle, you have Hunter-Schreger bands, and you have knurled enamel. So individually they can ask you as a 3-mark question, or they can ask you collectively under a name called as hypomineralized or hypocalcified structures of enamel. That is a frequently asked 5-mark question. Hypocalcified structures of enamel or hypomineralized structures of enamel. So among what you studied, what are the hypomineralized structures? That include enamel tuft, lamellae, spindle, all your 3 incremental lines, and rod sheath. So these are the hypomineralized structures which you see within the enamel. On the surface of the enamel, you can see 2 more hypomineralized structures, that is Nasmyth membrane or primary cuticle. Nasmyth's membrane or primary cuticle is a surface structure which is hypomineralized or which is not at all mineralized. Otherwise, within the enamel, these are the structures which is seen which is less mineralized. Now, there is another structure which you can see within the enamel, that is the junction between the dentin and enamel. Now, dentino-enamel junction. So, basically, it is the junction between dentin and enamel. If you see the surface between dentin and enamel, you can see it is as scalloping. It is not a straight line. You can see there is concavity and convexity. So, basically, the dentino-enamel junction is scalloping, and you can see depressions or concavity on the surface of the dentin. The dentin surface is concave, and the rounded portions of the enamel is going to fit into the depression. If you see, the dentin surface is pitted and projections of the enamel is going to fit into the pitted area of dentin. So this will give you a very tight interlock between dentin and enamel. So this brings about more adhesion between dentin and enamel. And remember, in permanent dentition, it is scalloping. In deciduous dentition, it is little more straighter. So there is a difference in the DEJ between deciduous dentition and between permanent dentition. Permanent dentition, it is scalloping, while in deciduous dentition, it is straight. Now moving on to the other structures which you can see on the surface of enamel or in enamel. That is collectively called as surface structure. So we studied the basic structure of enamel, which is rod, interrod, and rod sheath. Then the other structures which you see inside enamel, that is the light microscopic structures, that include spindle, your knurled enamel, Hunter-Schreger bands, tuft, incremental lines. Okay, and we have some structures which is present on the surface of your enamel. Those are collectively called as surface structures. So among the surface structures, on the surface you can see aprismatic enamel or prismless enamel or rodless enamel. So we had already said that aprismatic enamel is found at the dentino-enamel junction as well as on the surface. The other structures, what you see is pericymata. So pericymata is also seen on the enamel surface. And if you take electron microscopic view of the surface of the enamel, you can see rod ends, you can see cracks, you can see enamel pits, enamel cap, enamel brooch. So this is seen under electron microscope. And you have another thing which I already told you, which is a hypomineralized layer or an organic matrix that is called as Nasmyth's membrane, which is covering the surface of enamel. Now looking more into detail of these surface structures, aprismatic enamel has been already discussed, so we'll go into other structures. That is, the first one is perikymata. Perikymata, if you see this, see the surface of the enamel, it has got small lines on the surface of enamel. So they are basically extension of the striae of Fretzius. So incremental line extension onto the surface is seen as a groove, which is called as your perikymata. So if you see here, this is your striae of Fretzius, and you see the surface here, it is a small groove. There is a small depression where the striae meets with the outer surface. So that is called as perikymata. And they usually run circumferentially across the enamel surface. So this is called as perikymata groove. And if you notice between two grooves or between two incremental lines on the surface, you can see an elevated area, a raised area. That is called as implication lines of pickerel, or it is a perikymata ridge. So you have a ridge, you have a groove, a ridge, a groove. So the ridge is perikymata ridge, or it is called as implication lines of pickerel. And the groove that you see is your perikymata groove, which is the external manifestation of striae of Fretzius, which appears as a depressed area on the surface of enamel. Okay. The other structures, like I said, are the electron microscopic structures. So you can only visualize these structures under the electron microscope. So that includes the ends of the rods. So the ends of the rods appear as a concave area on the surface, and it keeps varying. So it is more deeper. The concavity is going to be more deeper in the incisal edge or the occlusal edge. And as you go towards the cervical region, the rod ends become less shallower. Other things which you can see is enamel cap, which is an elevation like this, which is seen on the surface of an enamel. So elevation, there are two types. You have enamel cap and you have enamel broch. Both are elevation. There is a difference in the size. Broch is a larger elevation, while cap is a smaller elevation. Now how is that you get deposits like a small cap, how you get on the surface? So if you have some unmineralized debris, okay, unmineralized area on the surface of enamel, on top of that you will have mineralization occurring. So you will have a debris, some dirt on the surface of enamel, and on top of that you will have mineralization taking place, which will form an elevation. So cap is a smaller elevation, a broch is a larger elevation. This is how it looks. And you have enamel pit. Pit is where your ameloblast resides. Oh, it represents the ends of your ameloblast. On the surface, you can see small pits or depression. So this forms your electron microscopic view or structures of your electron microscopic structures of enamel. Then we came across another thing which is formed on the surface, that is called as a Nasmyth's membrane. Now this is a membrane or a complete organic matter which is present on the surface of the enamel. It is a membrane which crowns a newly formed enamel or a newly erupted enamel. You can see a membrane covering the crown of the enamel, that is to protect the enamel. So it borders the surface of enamel and it has two components. It is made up of two things. It has an acellular portion. That means you have an area which is without cell, which is called as a primary enamel cuticle. Okay, and you have another area which is called as a cellular portion, which is made up of cells, and that cellular portion is basically the reduced enamel epithelium. So you have an acellular portion which is just adjacent to the crown area, and above your acellular portion you have a cellular portion which is a reduced enamel epithelium. So two layers your Nasmyth's membrane is made up of: primary cuticle and reduced enamel epithelium. Now, primary cuticle is just adjacent to the crown of the teeth. It is a membrane And this is actually your basal lamina. The ameloblast and the reduced enamel epithelium will resecrete the basal lamina. So that is your primary enamel cuticle. And the reason or the function for this is to protect the surface of newly formed enamel from resorption. The last part of enamel is related to the age changes and the clinical consideration. What are the changes that occurs when the enamel ages? So if you see, we all know enamel is non-vital, it is acellular, it is avascular. So if enamel is lost, it cannot be formed again and it is incapable of regeneration. So as age advances, enamel gets lost slowly. So it becomes very thin in an older age individual. Along with that, you can have attrition and wear and tear of your occlusal surfaces or proximal surface of the enamel. So enamel will be worn out. You will have attrition, that is wearing away of your enamel surface. Now, when you have wear and tear of your enamel, what happens to the oral cavity or what happens to the crown? One, the crown height decreases because enamel forms so much of thickness. So when enamel is completely gone, the height of the crown decreases. Then you have flattening of all the surfaces. Your proximal condor, if you remember when you study the morphology, you have convexity, concavity in the mesial outline, distal outline is also concave. Similarly, your labial surface is convex, concave. So you know that there is a contour for the mesial distal outlines. So this outlines, the convexity, the concavity and all becomes flat and straight when you have wear and tear of your proximal surface. And we also studied on the surface, we have this external manifestation of striophretius. You have your perikymeta groove and your ridges. Even that gets flattened out. So, one of the changes that you get is it cannot regenerate. It becomes thin as age advances. Another thing is there is wear and tear of your enamel. So, you can have occlusal wear and you can have proximal wear, that is on the side. And because of the wear and tear, you get lesser dimension or height of the crown. The proximal sides will become flattened and also the perikymeta which is present on the surface is going to disappear. The other changes what you get is related to the color of the enamel. We know that the color depends upon the thickness of the enamel or the opacity of the enamel. So as age advances, we know that the enamel keeps wearing away, it becomes thin. So it will appear darker in color. It appears to be more yellowish in color. That is because the enamel is becoming very, very thin. So one is that it will appear very yellow in color. That is because of the reduction in the thickness of the enamel, and it is the underlying color of the dentin that is going to be reflected on the surface. Second thing is that you can have changes from your environment. Suppose the patient has a smoking habit, the person smokes, or drink some kind of Ayurvedic medicines, or eating some kind of foodstuffs which will change the color. The color will be changed by the bacteria. So all this will produce color on the surface of the enamel. So this is what happens to the color as age advances. Another thing which we can notice is reduction in the permeability. So when age increases, the enamel is a semi-permeable membrane. So the permeability of the enamel will keep reducing. The pores between the crystal, we know that the crystal has got some pores between them. The pore size will keep reducing and the size of the ions will keep increasing. So as age advances, the permeability reduces. Pores between crystal will reduce in size as well as water content is also going to reduce in amount. So as there is, you know that there is hydroxyl ion and water is a hydration cell. It forms a hydrating shell around the crystal. So with age, the hydration shell will keep reducing. Now, the last part of enamel is the clinical considerations. What you have to look when you're cutting an enamel in a patient or when you're taking care of damages within the enamel. Or what other things which you have to take care about the enamel. One is when you're doing a cavity preparation. So in second year, third year, you will be cutting cavity within the enamel. If you have caries, you have to remove the caries and build the walls or cut the walls in a smooth manner. So when you do that, remember you cannot leave sharp walls or some part of enamel walls which is just not smooth and straight. You cannot have a wall like that. Now if you have irregular wall, that will lead to secondary caries. That is, when you fill a cement and when you have an irregular wall, there will be seepage of saliva in between the cementum and in between the cement that you place and between the wall. So this will lead to entry of bacteria, it will lead to leakage, and finally it lead to secondary caries. Another thing which you have to remember is when you have deep enamel fissures on the surface. If you have deep fissures on the surface of the enamel, again it can lead to caries. Then your enamel lamellae, which are organic structures, which will help in the entry of microorganisms and also help in the progression of dental caries. Another thing which you have to remember is fluoride application. So it is always necessary that you use a fluoridated toothpaste, or it is better that even in children, if the fluoride content is less in their drinking water, or they are not they are not having adequate fluoride in their diet, then it's better that you give fluoride applications. So fluoride has a property to reduce the solubility of enamel. It makes the enamel acid resistant. So it produces a crystal, that is fluor hydroxyapatite crystal, which is resistant to acid decay by the microorganism. So it is able to protect the enamel a little bit more longer than the normal enamel. So a fluoride containing enamel will add more protection to the enamel. So other things which you can see is during the development, you can have changes occurring within the enamel. So if there is less amount of matrix, then you have a disease called as enamel hypoplasia. So if it is because of environmental change, you have environmental enamel hypoplasia. If it is hereditary, that is your X chromosome in amelogenins. If there is a defect in that, it lead to amelogenesis imperfecta. So it can be environmental or it can be related to genetic changes. And if your entire tooth is going to be affected, where you have thin enamel, thin dentin, then you call it as regional odontodysplasia. And also you can have discoloration of enamel if during the birth the mother takes in tetracycline, or when during the development of the teeth, if the child happens to eat tetracycline, you can have pigmentation of your teeth. So these are the changes which you can get during enamel formation. So remember, to cover with the gist of the chapter, we studied about the properties of enamel, the physical properties related to the hardness, the solubility, the reflective property of enamel, the thickness of enamel, the color of enamel. So these were the physical property. The chemical composition of enamel, which had organic, inorganic and water. And in detail we read about inorganic matrix, what is the use of water and what is present in organic matrix. So organic matrix, remember, it is enamel proteins divided into amelogenins and non-amelogenins. A little bit detail about non-amelogenins and their role in mineralization. Then about inorganic matrix, you know it is hydroxyapatite crystals. Okay, what is an unit cell? What is the formula of an unit cell? And we went into this basic structure of enamel, that is enamel rod, interrod and interprismatic substance. What is enamel rod? What is it made up of? How are the crystals arranged? How is the direction of the rod? How is it different in deciduous and permanent dentition? What is the shape of the rod? What is the pattern of arrangement? The length, the diameter, all that we read under enamel rod. Again, then we read about what is interrod substance and what is your rod sheath, which is an organic matrix. Also, we covered on the structures which were present in the enamel other than the main structures. That is your light microscopic structures. And one question which can be asked is, remember, it is hypocalcified structures of enamel. So individually they can ask you light microscopic structures, or it can be hypomineralized structures of enamel. We discussed on what is aprismatic enamel. Okay, that again can be asked for 3-mark question. And then the surface structures of enamel, what you see on the surface of the enamel, that is perikymata, ultra structures, what you see under the electron microscope on the surface. You see broach, you see pit, cap, as well as enamel rod ends. And then Nasmyth's membrane. And finally, we went into the clinical concentrations and age changes. So this covers your entire enamel. Thank you and keep learning. All the best for your exams.